Hi, my name is Allison Drees. I teach at Southeast Polk High School in Des Moines, Iowa. And during the last MINK session led by Nebraska, I presented how I've been using phenomena in my classroom. And the one specifically that I'm going to share is for my cell cycle unit. And it stemmed from one of my students. So in the first phenomenon session with MINK that Nebraska hosted, they had a guest speaker on who was in charge of the NGS's Phenomena website. And I pulled a couple things away from him, which led me into the phenomena I picked. And one thing he said was that you should go with what your kids are wondering. And so this one came directly from a student who, for some reason, was talking about her little brother and how he got a gecko bit onto the end of his nose. And when he freaked out and went to pull the gecko off, the tail came off and it was still like withering on the ground, and it scared the bejesus out of her. And so I knew we were coming up to the cell cycle and mitosis, and I thought, okay, so my kids already know about this, and a couple of them are mentioning that they like had lizards as pets or geckos as pets. And so I thought, okay, they can already relate to this. That's a good phenomenon. And then the other thing I took away from that guest speaker was your phenomenon don't have to be anything crazy, ridiculous, over the top. So I wanted to keep it super simple. And that's where this came from. So I'm going to share my screen now. Oh, there we go. Okay, so this is actually what I gave them. Their phenomena was just this. I got this from Science News magazine, I think it's called. And this is the picture that they were using and the little comment. So it simply says, salamanders such as the axolotl above can regrow nearly perfect tails, including missing nerve cells. Lizards like the green anole below can't. And that's all I gave the kids to start. Then I showed them a little video clip here, but only the first few minutes of it so that it wasn't crazy long. I think I went only to about, a minute into it or so. So, okay, loading ad. So I'll go ahead and ignore that if I can actually get out of it here. All right, well, I'll just click into a different tab. Hopefully that's not creating any weird sound and stuff. Okay, so based on that really short video clip and the picture with the caption that I read out loud to them since it was projected on the whiteboard, I had my kids do a QFT protocol where they asked as many questions as they possibly could about the phenomenon. So I just had individual whiteboards and they wrote as many questions as they possibly could as they were doing that, and give them each 2 sticky notes. And then on the sticky notes, they had to pick 2 questions that they felt best would get us, if answered, to understanding the phenomenon. So basically just 2 questions that, if answered, would help us best figure out the answer. So these are some of their questions. Okay, after the initial one, we read out all of our questions. The kids identified patterns or trends they heard in the questions. So these categories you're seeing, like application, regeneration, death, cells, tissues, and organs, these are categories they came up with after hearing each other's questions. I would write those categories on the board as they said them, and then they came up and stuck their questions, sticky notes, where they felt they fit best. And you can see I had 3 different classes, so they came up with all sorts of different questions in the 8th hour. I just ran out of time to type those up and get them on. But so the next step was, how do I take this phenomenon and keep it going throughout the whole unit so that kids are constantly referring back to it? Because my one thing is I'm always afraid people think a phenomenon is just a hook that shows up at the beginning and then is never talked about again. And the idea of a good phenomenon is that it permeates throughout the whole unit and can be that anchoring point for all of the instruction and activities. Oh, great, there it's working. Okay, so the first thing I had them do was I had them look at a common phenomenon. So I pulled in something else, an onion root, and I said, an onion root cells undergo the same thing the axolotls does in order to make this regeneration possible. So we can compare these two species and the overall process they are going through to help us explain the salamander's special abilities of regeneration. So what I had them do was an inquiry-based activity where they were given this microscope image of an onion root tip with the cells undergoing mitosis, and they had to pick 6 cells. The DNA was doing something completely different or looked completely different. So for example, like I'm gonna try to circle over, like the DNA in this cell looks completely different from the DNA in this cell. So they had to look for 6 completely different looking cells and place them in a logical chronological order. So if, for example, if they could watch one of those over time, put those 6 in order of time. And that got them to the idea of, well, so an onion root tip, it's going to be growing. I had to talk them to that. And so if it's going to be growing, logically, it's going to have to start with a single cell and grow into 2 or create 2. That's the idea of the cell cycle mitosis and growth. So maybe that's how the axolotl tail starts to grow back. And so they linked it back to the initial phenomenon. Then I needed them to understand how the parent and daughter cells related. So we did a CER where I gave them this model, and they had to come up with an answer to the question to claim. And they did get to the fact that the parent cell and the daughter cells are genetically identical, completely the same. And so that led to more questions. So you'll see if I go back to the driving phenomena list here, it says questions after task 2, parent-daughter cell relationship. So this is where differentiation really started to show up as a category, because they recognized, okay, if the DNA in the parent and the daughter cells is exactly the same, they're genetically identical, then how is the axolotl growing back muscle and bone and skin? Because those cells are not the same, they're not identical. And so they didn't understand how you could have identical DNA, but different expressed genetics. So they were asking me all sorts of questions about differentiation after that. They also started talking about cell signaling, like how does a cell know what to grow back? Like how does the body even know muscle needs to be regrown? And errors and mutations started coming up. Well, what if these cells are splitting too much? Like what if they go too fast? So some kids actually were linking this back to cancer, things like that. So it just generated more questions. Okay, from those questions then, that's how I started creating my activities, because now we had such good, rich, in-depth questions driving us. I was able to take those and use them as formative assessments. And that's an idea I got from Next Gen Storylines, Brian Reiser's group. And he once said at an NSTA conference that they would have kids as formative assessments just go pull a driving question sticky note off the board and answer it after that day was over or that lesson was over. And so I thought, okay, well, yeah, they're asking the questions. Why don't I actually have them answer what they're asking? So we went more in depth with the cell cycle reading. We did some model interpretation, and then you'll see here in highlighted yellow, these are their questions that they generated from the previous activity. So I said, okay, after doing this reading and this learning or analyzing this model, pick one of these driving questions and answer it. And so you can see that that just went throughout more models, more reading, pick one driving question and answer it. Hi, Jed. So it went all the way through like that. I had a video embedded, things like that. And from that, then we generated more questions. I had a lot less that second time. So you can see down here, questions after task 3, very few and far between. But if I had said what questions still remain unanswered, I'm sure I still would have gotten a ton about that regeneration idea, because that still did not help explain why we were getting those different tissues. So then, after we came back from spring break, I presented them with another image from the same article I got the initial phenomenon. So this one, you can see the lizard's tail and the salamander's tail before growth on the left, before being cut off, and after. You can see that the lizard's tail is all cartilage and bone, or fat. I think it might be fat, the blue there. All cartilage and maybe fat once it's regrown, but the axolotl, the salamander, is actually able to regrow all three types of tissues: cartilage, nervous and red, and possibly fat and blue. That's my thought anyway. And so using that phenomenon, okay, kind of like reinforcing the first one, I again used that idea of, okay, here are these questions you've been asking me. We're gonna chunk apart our content, and after a chunk, you're gonna pick one of these questions and answer it based on what you've learned. So we dove into stem cells, we looked at different gene expression within stem cells, and we answered one of their driving questions that they wrote. Then we compared stem cells within humans versus planaria, which we had also cut open as another phenomena, a similar one to the axolotl. And we dove into the axolotl with a little bit of reading, went back and answered questions. They were curious about cell signaling, so I pulled in some resources for that. Again, went back and answered the question. And lastly, there was one about scar tissue. So this is above and beyond the scope of this standard, but my class is an advanced class, so we are able to go above and beyond the scope of the standard. If I were doing this in a general biology class, not an advanced one, I would stop the questioning and stop the learning and construction of knowledge before we ever even got to cell signaling. So I literally would have stopped it probably here with that comparison between organisms. That would be enough for general bio. And so I would determine that just based on the parameters of what the standard said is too much or too little. So I guess in general, the big takeaways I want to get across is that the phenomenon isn't a one-time thing. Okay? It shouldn't just be a hook. It should be permeating throughout the whole lesson or whole unit so that the kids have that anchoring idea to constantly go back to and wrap their learning around. That's just one example of how I did it. It might not be the best way to do it. I'm sure there are better ways out there, but that's as inquiry-based as I could make it to keep the kids invested by answering their own questions and frequently just going back in and asking even more. Our assessment was great. It was made by one of the student teachers here. She aligned it very well to what the standard was asking. And our kids had a free response question, essentially, where we asked them just simply, why can these organisms like planaria or the axolotl, why can they regenerate a whole limb when you cannot? And so our kids just free wrote about that using all the academic vocabulary they could and the content knowledge, and the products for that were absolutely outstanding. They were able to go above and beyond and pull in some of that extra stuff like the cell signaling or the idea of pluripotent stem cells versus somatic stem cells in humans. And that's it.